So we were talking about Weeks Bay and May's research on the factors that affect eutrophication in Weeks Bay, and so can you briefly explain the eutrophication and the factors in Weeks Bay?
Sure. So I have this map here of Mobile Bays, the big light portion here, and then you can see Weeks Bay is in the southeastern portion of Mobile Bay, and you can see how small it is. It's in Baldwin County there. So we looked at an 18-year time series of chlorophyll and nutrient salinity at Weeks Bay. It's a really great opportunity because Weeks Bay is part of the National Estuarine Research Reserve System, and so they have this really great data series from their monitoring programs. And what we found was that temperature, phosphorus, and river discharge were big components of how phytoplankton would change in the system. And it's really interesting because Weeks Bay actually shows the opposite pattern of river discharge as other coastal places that have eutrophication. So do you think I should rehash this?
Yeah, so as contrasted with the Mississippi River, which gets a lot of nutrient input, the Gulf of Mexico, which gets a lot of nutrient input from the Mississippi River, flowing through the breadbasket with a lot of agriculture. As you said, Weeks Bay is getting a lot of nutrient input rather from groundwater.
Yes. So when you have high river discharge, it actually dilutes the amount of nutrients that are entering Weeks Bay. And then also because Weeks Bay is so small, the river discharge, when it's high, it actually flushes out the entire system. So the phytoplankton aren't even there when the river discharge is high. So when there's low river discharge, so like drought times or low rainfall, then the phytoplankton actually have time to grow and accumulate. So that's a really interesting pattern that's really different from what we're seeing that results in the dead zone in Gulf of Mexico.
So the phytoplankton are little single-celled algae, and they are fed by nutrients. And sometimes the nutrients that are flowing from the land through these watersheds, these river systems, cause excess growth in the phytoplankton. And although phytoplankton produce over 50% of the oxygen on the planet, and they are the base of marine and aquatic food chains, so they're very, very important. When there's an excess growth and then a sudden die-off, they'll hit a different kind of limiting factor. And there's a sudden die-off, then the bacteria consume the phytoplankton, and when the bacteria consume phytoplankton, they consume the oxygen. Then you get these low oxygen areas. And so May has been looking at this particular area, Weeks Bay, and they have found that it kind of exhibits some different characteristics than a lot of the dead zones like in the Gulf of Mexico and the jubilees that are experienced up in the northeastern part of Mobile Bay. So when you guys, what are you looking for? Are you sampling phytoplankton?
Yeah, so I did the analysis of the time series, and like I said, we found variables that helped explain what was going on, but we also had a huge chunk of variability that we actually can't explain. So I'm going to tackle that by doing a field study. I'm actually almost done with it. It's going to be finished up in July. And that field study focuses on looking at growth rates of phytoplankton and grazing rates. So how fast are these phytoplankton actually being eaten by grazers in the water? And hopefully that'll help us answer a few more questions.
So the grazers would be little small zooplankton, animal plankton?
Yes.
So when you go out into the field, you go out into the field and take samples?
Yes.
And so you'd be sampling temperature and taking maybe looking at rainfall patterns.
Yeah, so they actually have a meteorological station set up. So that rainfall and light and all of that is already taken care of by Weeks Bay. So they do that monitoring, and they also do continuous monitoring for salinity and temperature and oxygen and pH. So that's some great foundation that I have. When I go out, I do also just take salinity, temperature, DO, all of those things, just to make sure I have them. And then I actually take like big 20-liter jugs of water at each of the stations that I'm sampling. And then I bring it back to the lab, I set up my experiment, and then I measure growth and grazing rates from those big jugs.
So in your big jugs, you have collected phytoplankton and zooplankton.
Yes.
And then so you kind of set up a captive closed system here in the lab.
Yep, I set it up. I have like 36 bottles, and then I throw them in our mesocosms and let them grow for 2 days, and then I come back and measure how much they grew over that time period.
So give them kind of like 2 days worth of growth and grazing, and then look. What do? You do like what is it that you're measuring? Are you looking at the population in there? Are you looking at?
So I do. I have been looking at the population. We have a flow cam in Dr. Krause's lab, and so I take samples of the water and then we run it through the flow cam, and it takes pictures of everything that's in there. So in that way we can find what kind of phytoplankton specifically are in there.
Wow.
Yeah, and then we also, the growth is measured by how much chlorophyll is produced. So we have these filter systems here. So basically what we do is we take these filter pads, these little filter pads, and then we put them on here. We screw in this top, and then we just pour the water in here and we pump it out, and then what's ever left on the pad, we extract the chlorophyll from that. So what that ends up looking like is you have this pad that was full, and then we put methanol in there and we shake it to bust it up, and then we let it freeze for a day, and then we use the fluorometer that measures how much chlorophyll is in there. So yeah, that's how we get the chlorophyll measures.
And then how do you estimate the grazing?
We estimate the grazing. So in order to get that, what we do is we make dilutions of the full strength seawater, and then we add in water that doesn't have any phytoplankton in it. And so based on the curve of the dilution, we can figure out what the grazing rate is.
And so are you, what kind of like seasonal patterns are you finding?
Yeah, so the seasonal patterns, we saw seasonal patterns in our time series analysis. So in September is when we have the biggest boom of growth. So basically, if you have a graph of January to December, it's really low in the winter months, and then kind of goes up a little bit in the spring, and then in the summer, and then right at the end of the summer to the fall, you have a big boom, and then it goes down again. So yeah, a lot of, the canonical spring bloom that you see in the Atlantic, we don't have that here in Weeks Bay. We have a summer to fall bloom.
So would this seasonal pattern be affected by things like rainfall, so the water input, and then also temperature, because these are, photosynthetic organisms that are, dependent on sunlight.
Yeah.
And you did mention that there's, flushing when the river system flushes. And so that pattern is a little different than you see with some hypoxia. But still, when you would maybe expect to see the more groundwater flowing in, is there a lag between the rain and the groundwater flow into the system?
I'm not sure about that. I haven't looked specifically at that, but the seasonal patterns, they are affected by all the variables that you mentioned, but the biggest one is temperature. So you will see that boom is mainly due to temperature. Another reason it's good to look at these long time series is because there's so much noise in the data. So like you said, you might have a flushing event. You might have one day where there was no sun, and so that day was littler growth. So when you look at these series, on a big scale, then you can kind of filter out some of that noise and see what is affecting the phytoplankton the most. So the ones that we found were temperature, phosphorus, and river discharge, plus some other ones that we don't know what they are. So there's plenty of room to learn more about what's affecting them. But yeah, see the seasonal patterns, that's going to be climate for you. Affecting the phytoplankton.
So you earlier mentioned nitrogen as well as phosphorus. Why do you think you're seeing more of an effect from the phosphorus in this system?
Well, that's because the system actually isn't nitrogen limited. So normally what you have is freshwater systems are phosphorus limited, and then marine systems are nitrogen limited. But because this system is, number one, is actually very fresh, and number two, we have such high amounts of nitrogen that there's actually completely sufficient nitrogen for the phytoplankton to use.
Okay, so let's just clarify what we mean by like these different nutrient limitations. So if a plant is, or an ecosystem is phosphorus limited, that means that under normal conditions, normal environmental conditions, that would be like a limiting factor for the growth of the plants or the algae. And then if you put a lot more phosphorus in, then that is no longer a limiting factor for their growth.
Yes.
So that's what we mean when we say that. So under normal conditions in Weeks Bay, there's plenty of nitrogen.
There's plenty of nitrogen, yeah.
So that's not a limiting factor for their growth. It's a different nutrient that's a limiting factor for their growth. So the nitrogen input doesn't cause noticeable change.
Yeah. So although that is something we should control because the nitrogen levels are extremely high, actually controlling the phosphorus would probably have a better effect. Because it's the phosphorus that is really limiting how much can grow.
So what are some of the other limiting factors that the big bloom, and sometimes we call these blooms, but I like to point out that it just means like a lot of proliferation of the algae. There's no actual like flower bloom associated with them.
Yeah.
But what would be some of the other limiting factors that they might hit that would cause a big die-off?
Let's see. Well, like I said, temperature for sure. A big die-off. Grazers. So if you had, when you have a lot of the phytoplankton, you are creating all of that food for your zooplankton. So maybe your zooplankton just grow up so much that they actually just consume all of that phytoplankton. So that's another way that you could have the phytoplankton go down. And then, a big one would be a flushing event that would just push it all out.
So I always like to point out to people that, we do basic research, which is we just have questions about, what is happening, with different things. And so there's research to answer questions. And then sometimes that leads to, applied research where it has some sort of practical application. So they both have value. So I think, a lot of times people question the value of basic research, just kind of answering questions that at the time may not necessarily have some kind of practical outcome in mind. But they're kind of pieces of a puzzle. So when you get enough pieces of that puzzle, you can resolve some questions that may then lead to some sort of practical application. So this is an issue that people contribute to. So we are contributing to problems in these ecosystems by putting excess nutrients in the water. So we may address these problems that we're creating. And they're problems not just for the ecosystems, but they can have impacts to, humans as well.
Yes.
So what might be a practical outcome of this?
Well, the research that I'm doing is definitely extremely related to practical science. It's basic research, but what I'm hoping it can do is help inform management decisions. So we've known that nutrients cause eutrophication for a really long time, but sometimes it's harder to see what the impacts of all those interactions are. For example, in Weeks Bay, before I completed this project, I wasn't totally sure if nutrients were the main cause of the eutrophication or we're seeing some hydrology interactions. Now we're seeing that phosphorus is definitely not only a main cause of the eutrophication in Weeks Bay, just like nutrients always have been, but also that phosphorus is continuing to rise in the system, even though people have been trying to do nutrient reduction programs. There's many state, federal organizations that have been trying to do nutrient reduction programs. So it's kind of, just another push to be like, hey, well, maybe these programs aren't working. Maybe we need to figure out something else. And then also, I don't remember what my also was.
Have you identified some sources of the phosphorus?
I have not looked at that. There's been other people who have, and it's some municipal sources, but mainly they think it's from agriculture because Baldwin County is has so much agriculture. But actually, I may be wrong to say that because agriculture, it's about, right now, what is it? It's about 50% of the land is agriculture, and developed land has actually been going up. So agriculture has been going down, but we've been seeing an increase in developed land. So in the 70s and the 80s, it was maybe like 3%. Now it's in the mid-teens. And that decrease in agriculture, I think we all hoped it would decrease the amount of nutrients. But in fact, it seems like the increase in developed land has more than accounted for any decreases that we would see from agriculture. So I think that's something that we really have to focus on when we're doing more development.
So there's still questions about that. So, I'm just throwing out an idea here, but with development, often there is land clearing,
Yes.
clearing vegetation from the land. And that changes the way water flows across the lands, and seeps into the ground and moves. It changes the way water moves. And that vegetation also acts as a filter.
Right.
So it could be that just a question.
Yeah.
Not that there's evidence that this is what's happening in Baldwin County, but
Yeah.
Maybe the water is moving differently because of all of the development, and it wouldn't necessarily tell you the source of the phosphorus, but it might tell you something about why the phosphorus is getting into the
and I'm sure other researchers have asked this question in Baldwin County. It's just not specifically what I've done, but for sure we know that increase in developed land does the things that you said, the taking away the filter vegetation, increasing impervious surfaces so everything runs in, increasing just waste in general, increasing home fertilizer, which has actually been shown to be a cause of eutrophication. Just home fertilizer, not even just agricultural fertilizer. So I think it's really important to make sure that we keep on top of our monitoring so that we don't, let loose and say, okay, well, let's just do this development and not keep in mind that we need to also implement strategies to reduce nutrient pollution while still being able to grow our cities.
So did you have something to...
Yeah, so my study right now is looking at phytoplankton growth rates, but it's also important to know how much energy is being produced from phytoplankton, and you can actually do that with a very simple bottle experiment. So I set one up for us today. I have these, this is just a replicate, so we'll just put this one aside for now. I have these two bottles, and I took water from the dock at the Sea Lab yesterday, and I measured how much oxygen was in this water. So today what we're gonna do is measure how much oxygen is in the water after these guys have sat outside and grown in the sun for a while. And because we know that photosynthesis creates oxygen, we can then take that difference in oxygen and then calculate how much energy has been produced. And I did a little twist on that since we're talking about eutrophication today. So I actually added nitrogen and phosphorus into this one with the green tape. So this bottle represents our system that's undergoing eutrophication, so it's enriched in nutrients. And so what we would expect to see is that oxygen will have increased in both of these bottles, but that this bottle may have more oxygen than this one because of the nutrients that cause more phytoplankton growth. So I have this oxygen probe here, and it's a awesome, very simple instrument.
So real quickly, let me just ask this question, show the map. So this is Dauphin Island right here. So this is where you took the sample, right over here on the east end of Dauphin Island. Is the plankton, the species of algae that you see here, is the group of algae that you see here similar to what you see in Weeks Bay?
So it should be pretty similar just because of how close it is in location, but the salinity of Weeks Bay is really low. So the average salinity is maybe like 5 parts per thousand, whereas in Dauphin Island we get all the way up to full strength, and it obviously comes there's a cycle with the tides and everything. But I would expect to see more marine phytoplankton here than here in a system that's so freshwater dominated. So let's take a look at this. So yesterday when I tested the water, you can see right here we have % oxygen saturation, and 100% would be what the air is. So yesterday when I tested these bottles, they were at about 70%, and we can see that this bottle has now increased to about 74%.
So one of the things that I also like to point out, just because I think some people have some confusion about this, when we talk about oxygen in the water, we're not talking about the water molecule that people know as H2O.
Yes.
It is O2, oxygen gas, that is held among the water molecules. So it is, we say it's dissolved into the water.
Yeah.
But these molecules are kind of, the oxygen gas molecules are kind of held among the water molecules.
Yes. That is definitely something that can be confusing because, yeah, oxygen is in water, but it's in the water molecule. So we had about a 4% increase in oxygen for that one bottle that had no added nutrients, and let's see what we have for this bottle that had the added nutrients.
So there's a little rotor in there that's kind of stirring, is that to kind of homogenize the oxygen?
Homogenize, and then also the probe works by the oxygen actually dissolves through the membrane, and then it sends an electrical signal, so it needs like the oxygen to be continuously moving over the probe.
Okay.
Yeah. And it looks like we're getting to be stabilized. So this bottle has 83.4%. So we can see even from just a day of growth with increased nutrients, this bottle had 10% more growth than the other one, or more oxygen than the other one.
So the oxygen represents the growth of the phytoplankton.
Yeah.
And, and then again, because we're relating, put this reminder out there that when you've got excess nutrients flowing into a system, you have an initial increase in oxygen, and then you have a plankton die-off, and that then results in a steep decline in oxygen.
It can, yes. Yeah. We don't actually see it that much in Weeks Bay, but we're not it's not that huge mass that the Gulf gets. Yeah.
And then do you want to talk just real briefly about the Jubilees, because there's so much interest in Jubilees?
Yeah. I don't know that much about Jubilees, but I know that they've been occurring regularly in Mobile Bay. I think, I don't even, how long, Mendel? Like, they're in papers from early days. So it is a naturally occurring process. And like I said, it's usually pretty fast, like, it'll happen for like a day or so. So I believe that it's not actually caused by eutrophication, but that it's caused by physical factors like how the wind is blowing. If the wind is blowing some warm, salty water in, it can set up stratification, which means that the layers in the water are separated. So it's that same thing where because the layers become separated, that water can't mix up and get oxygenated. So don't quote me on that, but I think that's what they think jubilees might come from. And the problem is we do have hypoxia from eutrophication in Mobile Bay. Well, we don't know for sure, but we think that that might be happening too. So what happens is if you have this like physically forced hypoxia and then you add the biologically forced hypoxia, then you could make the low oxygen even worse.
So typically when there's a jubilee, you've got sort of a wall of low oxygen water that's being pushed towards shore. And so there are animals that are fleeing from that, shrimp, crabs, flounders, eels, that are kind of fleeing from that low oxygen area towards, they're in search of, trying to handle the camera and look.
Yeah.
I pointed the wrong spot. They're in search of oxygen, so they're kind of being they're fleeing from this low oxygen area, and they will sometimes go into very shallow water, kind of right up on shore, and people will go and scoop them up. And so it's been called a jubilee because people are
great for fishermen, but
excited
then when you know
to go scoop up a lot
what the
of
fish
seafood.
are doing, it's a little morbid.
So is there kind of a big picture message that you'd like to leave folks with about hypoxia, about eutrophication, about the need for research and regulation?
I think the big picture message is just something that we've been saying for a long time. My research is not novel in the fact that we're finding that nutrients have a big impact on eutrophication and phytoplankton growth. But clearly it's an issue that we can't get rid of, and it's only getting worse. So just to, be aware that as humans, we really can impact what's going on around us. And like I said, even small things around your house and when you're fertilizing your yard, don't use more than you need. Make sure to always put waste where it belongs and not just to let it out, because all of that will end up in our water.
And then, things like using pervious surfaces in
absolutely
developments that allows
try not to pave everything. Maybe plant like you said, use gravel instead of concrete.
And these, what you're doing is kind of improving the resolution on this. This what we understand about what's going on in Weeks Bay, and then these can be applied to bigger scale pictures. So with that fine tuning of resolution with these ongoing questions. So there's still a lot of questions about what's going on with.
And because we know that eutrophication is affected by climate, and we are in a changing climate right now, a lot of the biggest questions people have is, how is climate change going to affect eutrophication? Like I talked about, river discharge is inherently linked to climate. So more droughts, more storms, higher severe rainfall, all of those things will come back to this. So that's something that a lot of people are working on now, to try to understand what these factors are doing before they change, and then hopefully be able to predict how they're going to change our environment.
Well, thank you so much for chatting with us this
Mendel.
morning, and I'm sorry for the technical difficulties at the beginning. The Angela, who usually handles our camera and production, was unavailable this morning, so I was trying to do both and made a mistake. But thanks for joining us, and thank you for explaining this to us. And we all look forward to understanding this better.
Yeah, me too. Thank you.